The overall goal of this procedure is to label and visualize mtDNA replication in cultured neurons. This is accomplished by first incubating neurons with the thymidine analog EDU for 2 to 24 hours. The second step of the procedure is to label the EDU that contains an alkyne with the fluorescent Oregon green 488 azide through a copper-catalyzed covalent reaction. The final step of the procedure is to amplify the Oregon green signal with a tyramide signal amplification step in order to visualize the EDU that was incorporated in newly synthesized mtDNA. Ultimately, results can be obtained that show mtDNA replication in specific subcellular compartments of neurons through the combination of click-it EDU chemistry and subsequent tyramide signal amplification.
The main advantage of this technique over existing methods like BRDU labeling is that EDU labeling is easier and milder, allowing for additional immunofluorescent staining of neuronal markers.
This method can answer key questions in the regulation of mitochondria number, such as how neurons meet changing energy loads in their subcellular compartments, including cell bodies, axons, dendrites, and synaptic terminals.
So this method can provide insight into the mechanisms underlying the pathogenesis of multiple mitochondrial-based neurologic diseases, including neuropathy and neurodegeneration. But importantly, it can also be applied to other systems where changes in mitochondrial DNA copy likely underlie the pathogenesis of the disease state, including drug toxicity, cancer, and aging.
This video will demonstrate